This is the one we need to study in great detail, though. So Rhizopus stolonifer is the subject of most of this topic, okay? Yeah, so somebody's asking if you're asked for, yeah, an edible mushroom, button mushrooms. So yeah, they would, absolutely. So button mushrooms is fine to say as an example of edible ones. The ones they were looking for are the harmful thing. They wanted you to name a harmful mushroom, a poisonous mushroom. So you had to say something like the death cap or the destroying angel or that red and white, that typical, like, you know, fairy tale red and white spotted mushroom. That's the fly agaric, it's called. Fly agaric, you might know that already. So no one name, okay? Just to cover yourself. Of course, now they've asked it last year. What are the chances of asking it this year? Fairly low. Okay, so here's the deal with Rhizopus. You know this because this is the mold that grows on bread. It's sort of pin mold. It produces these little tiny sort of gray hairs with little tiny black heads on them. We'll zoom in and look at it in a second, but I'll show you some pictures in a second. It's a saprophyte. It's a spoilage. It causes food spoilage. It's a decomposer. They reckon that about 50% of the world's food is lost, not to just to Rhizopus, but to fungal spoilage, right? So rotting. Things rot because of these guys and others like them. Bacteria and fungi. Fungi are responsible for an awful lot of it. So, you know, mold, right? Saprophytes is what we're talking about. This is a saprophytic bread mold. It does what I said, secretes extracellular digestive enzymes onto the bread and absorbs the digested products. The hyphae then are for two things. They produce the digestive enzymes, and they're also involved in the absorption of the products. Hyphae are these little very, 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 very hair-like thin threads that we were talking about that make up this mycelial mat. So it will just keep growing. And it's a funny thing about mushrooms. They reckon that you know, you could argue that fungi are the largest organisms in the world. So there have been cases where they've tracked a mycelium that seems to belong to, you know, one continual network of threads that has gone for a 50-mile radius or more. And the mushrooms are just what we see poking out of the ground every now and again. So what we look at called mushrooms are the reproductive structures, and underneath the ground is where all the action is really. That's where the mushroom really, that's where the fungus really lives. So, you know, it goes on for a network of often for miles. So this is a black and white picture. I'm choosing this because it's kind of easy to draw, and that's what I'm big into, making sure that the pictures that are in the notes are simple, two-dimensional, reproducible in an exam, quick and easy to draw, right? So one thing I'll warn you about, and we'll do some drawing when it comes to plants in particular. I'm going to use that as our opportunity to showcase how we draw stuff. There's a problem with this drawing, and it's that because I just lifted it from somewhere, is that you see those arrows that say sporangium is pointing in the general direction of a sporangium. That's really not the way you should be labeling things. The way you label things is you don't use an arrow. You use a sharp pencil, you put your ruler on the page, and you touch the thing that you're labeling, and using your ruler, you draw a line away from it. This is the pointer, right? But it must touch the object that you're labeling, not just point in its general direction. So touch it, draw a line out to it, and out from it, and somewhere outside the diagram, you write the label. So that's a crucial skill when it comes to drawing. Keep your drawings definite lines, no shading, no 3D, no color, none of that stuff. Simple, you know, reasonable sized, a good gauge if you're looking for what should the size of a drawing be. The average drawing should be the size of the palm of your hand. That's just the drawing, and then the labels should outside that. If it's smaller than that, it's likely too small. If it gets too big, then it's going to take you too long to draw it, and it's more awkward and more difficult to draw it. Okay, so there's your guidelines. We'll come back to drawing again, but this would be a good chance for you to practice your drawing sometime during the week. Is there's various different drawing opportunities in these topics we'll be looking at today. So this is what we call the asexual reproductive. It's not just the asexual reproductive structure. This is the normal sort of structure, you know, of everyday life of a Rhizopus. Okay, so those rhizoids, think of those as like anchoring, almost like roots of plant. They're analogous to the roots of a plant. They're not roots, right? Because they have different functions, but they're in some ways they're similar because they do anchor the plant. They anchor the fungus. They anchor the rhizopus on the substrate, in the whatever's growing on. Okay, so that's one thing. Stolons are hyphae. So sorry, rhizoids are an example of a specialized hypha. Hyphae are the threads. Okay, and one type of hyphae are these rhizoids. Another type of hyphae is ones hyphae that grow usually above the surface of the substrate, what they're growing on, right? And they're called stolons. And if you know strawberries, if you ever grow strawberries or you've studied strawberries in plant in your class, strawberries produce these aerial stems, and they kind of grow out away from the plant, similar to this stolon, grows away from the rhizopus structure, above the substrate. And at some stage, though, usually just gravity will pull it back down again. It'll touch the substrate at some distance from the original rhizopus structure. And where it does that, it throws out more rhizoids. More rhizoids will grow into the substrate, anchoring it, and it could then grow another sporangium. So this is like how one way that it can grow asexually, right? It's literally just cloning itself by sending out a kind of a stolon, like a shoot, above the ground, above the substrate, that eventually will touch down somewhere close by or whatever, and then produce another bunch of specialized hyphae, like rhizoids. But what it also will produce is another type of specialized hypha called a sporangiophore. Now, a sporangiophores purpose is just to raise this structure that's on top of it called a sporangium. Sporangia are the spore-producing structures. This is the sporangium. It's a kind of a bulbous black. And these are almost microscopic. You can barely see these with the naked eye, but they're tiny, smaller than the head of a pin, right? So inside that you find thousands of these microscopic spores, and so there's a kind of a release system inside, like a kind of like a springboard or like something like that, and it'll propel the sporangia, the spores up into the air, and they get carried away by the wind and the rain and washed away and all that kind of stuff, right? So that's the classic kind of asexual reproductive structure is the sporangium producing the spores. That's the main asexual reproductive mode for Rhizopus. But like I said, stolons are also involved in doing some sort of asexual reproduction. So the deal is, the whole story here is mitosis. So go back to our cell division, our nuclear division story. This is all about mitosis. Mitosis produces, is what's responsible for the growth of this, right? So one cell becomes two cells, although calling them cells, nuclei is what we're really talking about here. The nuclei get, divide by mitosis, and you get two nuclei, and the more nuclei you get, then the more these structures can grow. Because here's the really weird thing about this is that this is just another version of that picture, but that's in your notes as well. So it's good to have just to see different versions so you get a clearer picture of what's going on. Okay, so the interesting thing about this is that the nuclei in these hyphae, they're not separated into individual cells. Like, this is the whole thing we were talking about. We consider cells to be these individual compartments of which all living things are made. Yeah, not quite. So fungi are kind of exceptional like this. So they produce these more like these filaments, these really, really narrow tubes, hyphae, and the nuclei kind of flow along them. They're not separated into individual compartments, right? Which is kind of weird. So, sorry, just let me just make sure there's nobody banging on the door here. Yeah, we're good. Okay, so that's the asexual reproductive structure. This is what you're looking at when you're looking at it under an electron microscope. On the right-hand side is just, that's not a microscopic thing, but you can see the tiny little black heads on these gray threads. Some of the gray threads are just mycelia, regular stolons and whatever, but some of them are aerial hyphae, which are the sporangiophores. And on the little black pinheads are the sporangia, right? Each one is a sporangium. And inside those, you get lots and lots of microscopic spores. So that's just a table. Oh, I'm big into tables. You might have seen in the notes, there's a lot of tables. Tables are useful, really useful when you're looking for structure-function relationships. And there's a great way of testing yourself for various, is to try and summarize things into a table. You know, say, and especially when we come to, you know, like structure-function relationships in the human systems. So if you're talking about things like, you know, like the neuron or the structure of the leaf, or different, so organs and plant and animal organs, you know, the digestive system. Each part has a role and has a function. So it's a really good way to summarize. Pull yourself up a table, draw yourself out a quick table. See if you can make a list of all the important parts of this, whatever it is you're studying, and see if you can assign a function or two to each of them. It's a really good way of checking your learning.